Okay, can you see that slide?
The one that reads with seed dormant.
Yes.
Yeah, I can see it.
Okay, so today we are going to talk about weed seed dormancy.
Mm.
So, what is seed dormancy? It is actually a state in which viable seeds, spores, or buds fail to germinate even when exposed under the conditions which are favorable in terms of moisture, temperature, and oxygen. It is the failure of viable seed to germinate under conditions favorable for growth of the seedling. That is the general understanding of weed seed dormancy. Here I've got Datura stramonium seeds. Now, for them to germinate, dormancy has to be broken first so that you have this germination process, which simply is the protrusion of first the radicle, then followed by the plumule. You can see that short radical. It's a sign of germination. So that is the germination process. If we examine the internal structure of the weed seed of the seeds, you may find that they already have inside them radicals. They also possess cotyledons. Please remember that it is the cotyledons which store uh, food or food reserves, which are going to be used by the uh, by this radical in order to get outside the seed, or the food reserves will drive the germination process. And outside the seeds, there's this tester which has to be broken by the radicals as they germinate. So, what is the seed germination process? There are three things. Germination is the resumption of embryo growth and emergence of the seedling from the seed. I'm sure we are quite familiar with this process. It's the process of initiating growth in seeds. C, it is the resumption of growth of a seed or any vegetative part. The process requires moisture, one, number two, oxygen, three, temperature, optimum temperature, and sometimes it may require light in the case of photoblastic seeds. We can have live seeds in the soil, but they will not germinate. These seeds are said to be dormant. So the seeds which will not germinate in the presence of favorable conditions are considered to be dormant. There are types of domains which were actually described by Harper in 1975. He stated that there are three types of domains. You may have some seeds which are born dormant. If you have seeds having already in possession of dormancy, as they are released from the plant, it is known as innate or primary dormancy. Then some seeds may acquire dormancy after the release from the plant, whilst they are in the field or in the soil. This is known as induced dormancy. Then some seeds may have to acquire, uh, may have dormancy which is forced upon them by certain conditions. This is known as enforced dormancy. So there are three types of dormancy. The first one is innate or primary dormancy. The second one is induced dormancy. And the last one is enforced dormancy. So induced and enforced dormancy, if you like, you can call them secondary dormancy, as opposed to primary dormancy, which is innate dormancy. We can use a schematic representation of the types of dormancy. So let us define what is seed dormancy. First, in fact, in some literature, they refer to secondary dormancy and primary dormancy. Now, primary dormancy is inborn dormancy or innate dormancy. But secondary dormancy can be divided into two groups. You can have induced dormancy as well as enforced dormancy. Later on, we'll explain these three terms: innate dormancy, induced dormancy, and then enforced dormancy. So seeds from a single weed species may actually exhibit one or more types of dormancy, or all three in succession over a period of time, depending, of course, on the environmental conditions. So I'm going to use the dormancy status of a common ragweed, which was studied somewhere by Bazar's 1979. I just want to explain how weed seed dormancy can change over a period. On this diagram, I want you to notice that we have summer, autumn, winter, and spring. Now let us start with the summer. Summer is signaled by germination, which forms seedlings. These will become mature plants, go into a state of flowering. Then there is a seed dispersal. The seeds which are dispersed, they have already some domains, and that is known as innate domains. Then, when we approach winter, like this period in June, July, when the temperatures are very low, in some cases, the seeds are under freezing conditions. They undergo what is known as stratification. Stratification is subjecting seeds to low temperatures. What stratification does is that it can increase the ratio of promoter to inhibitor ratio. In other words, it can increase hormones which can later stimulate germination. For example, during stratification, you can have accumulation of gibberellic acid, which will later break dormancy. So, in spring, if we come in with a plow and disturb the soil, we expose the seeds to light, and the temperature may fluctuate, and the seeds are now exposed to low concentration of carbon dioxide. These conditions will break dormancy together with the hormones which have been built inside the seed. So germination will now commence in summer. It could be high germination of seeds, and the cycle will be repeated. Now, if we have undisturbed conditions where we are not disturbing carbon dioxide levels and light, we may have reduced germination following this route. So there could be low germination. And this is what we think should be happening in conservation agriculture. When we don't disturb the soil, we expect a low germination. Right? Let us look at the inner circle. Now, after stratification, you may have some seeds being forced to undergo enforced dormancy because there is no disturbance and there is no light, so they are under dark conditions. So they go into enforced dormancy. Then later on, they can go into induced dormancy, and they can keep on, changing status. On some occasions, they can follow this outer route or this other route. So the seeds are always, changing from either innate domains going into enforced, then induced, and sometimes they actually go into the germination process should the conditions allow. So this is what can happen to the weed seeds. So let us focus on the primary or innate domains. It is considered an inherent property of the seeds as they leave the parent plant. So when the mature plant releases some seeds, they already possess innate domains or primary domains. This type of dormancy is genetically controlled, and hence the period of dormancy may depend on environmental conditions. Later, I will explain about genetically controlled dormancy. These seeds will not germinate even when exposed to environmental favorable conditions for the germination to occur until their dormance period expires. Now, what causes primary or innate dormancy? There are a number of morphological and physiological factors that regulate primary or innate dormancy. Yeah, we are going to explore these now. Now, sometimes when seeds are shed from the plant, inside them we have got immature embryos. The embryo is still growing, even if the seed has been shed. So some weed species have the seeds which require an after ripening period for the embryo to mature. So sometimes we talk of this after ripening period, after weed seed shed. That period is necessary to allow the growth of immature embryos. Once they are mature, then the seed may commence to germinate. Now, some weed species may exhibit what is known as polymorphism, where they produce morphologically and physiologically distinct seeds which have different after ripening periods. For example, the Xanthium stramonium, which is cockroach. Now, this weed has two types of weed seeds, which are quite different. And this is what is known as the polymorphism. It's the condition where you have a plant producing different types of seeds, which are morphologically and physiologically different. You may have seeds which are born dormant or have innate dormancy. Uh, One of the examples is Acanthospermum hispidum. Now, when you have mature seeds of this chidongi, the seeds are already dormant when they are leaving the plant. The reason is that the embryo inside is not mature, but after the after ripening period, you have a mature embryo. Now, this is the Xanthium strumarium seed with these spines. Now, if you open up this case, you may find the two distinct seeds with the different domains characteristics. And this is what is.
Sorry. So, do you mean to say if we open this seed, you will find another two seeds inside?
Yes.
Ah. Oh, okay.
Yeah, I could have some seed at the crop science department. I will check.
Oh, okay. All right, it's okay.
Or I may have to. Yeah, I think I will have some samples.
Mm-hmm. Cool.
Yeah. So if you cut it, but can you see that it may be difficult to cut because of those
Yeah,
spines?
honestly, those spines will just make the work difficult
Yeah,
for you.
you open it after paying some price, a price, you know.
Yeah, something like that.
I have just been talking about immature embryos, which may make seeds fail to germinate during a certain period, which we call the after ripening period. Now, there is something which is known as embryo dormancy. Anyway, it's also embryo domains which I was talking about. But the only difference is that the other one, the embryo is not mature. But in this case, you have a mature embryo contained in the seed, failing to germinate or grow even when isolated from the seed or dispersal unit and exposed to conditions favorable for growth. So this kind of domains may be imposed by cotyledons. So if you want the seed to germinate, you may have to remove one of the cotyledons or a part of the cotyledons. Then the seed will break dormancy and germinate like this plant Fraxinus species. But some of our weed seeds, which we have in Zimbabwe, could have this type of dormancy. Probably we just need to make some investigations or to study dormancy in some of these weed seeds. Now, the other thing is that, let me go back. Here we said it's the cotyledons which are actually preventing the seed from germinating. That is a morphological impact. But you can have a physiological, suppression of domains under embryo domains. For example, seeds may produce ethylene, which stimulates germination of several weed seeds. Now, in a vinifatua or wild oat, ethylene levels increase just before germination. So, for germination to occur in a vinifatua, you need the presence of ethylene. Now, sometimes that ethylene is not sufficient, so you have dormants. So the seed may take a bit of time to build up this ethylene up to a certain level, which can then trigger germination by breaking dormants. The other substance these are actually hormonal hormones. Gibberellins, GA, these have been found to increase in those seeds that require chilling or stratification. Previously, I was talking about stratification under natural conditions. Uh, it simply means subjecting weed seeds to low temperatures so that the seeds may start producing gibberellins. And when these seeds are exposed to conditions suitable for germination, they have now acquired high levels of gibberellins, which will then cause germination. So GA stimulates, in short, I mean, it's gibberellin, stimulate degradation of food reserves in the endosperm or cotyledons, which is an important process in germination. Another hormone known as ABA, abscisic acid. Now, abscisic acid deficiency is associated with absence of primary domains. So, if the seeds do not contain, well, if the seeds contain low levels of ABA, it means they are going to be dormant. Now, high ABA content could promote seed, sorry, the absence of high levels of ABA will actually cause, I mean, the ABA deficiency will remove primary domains. But high ABA content could actually promote seed dormancy. So ABA is synthesized in the embryo and the endosperm. But what finally determines whether a seed is going to germinate or not is the balance of GA and ABA in the seed. This is quite a complicated process which was described in this paper. Probably later on you may have to follow it up. Seed dormancy and the control of germination. And we have this quite complex system where you have a seed which is dormant and you have got the ABA and GA during after ripening period, causing the seed to be non-dormant. And you may have light requirement and GA causing the germination process to be initiated. So you keep on having various, even ethylene is involved. Then finally you get your germinated seed. But from the dormant seed up to the germinated seed, it's quite a process. So if you wanted to explore this, you may have to read this paper.
So, Doc, does
Yes.
it mean to say that depending depending on the seed that we have, there is actually a different amount of any hormone that will trigger germination?
Yeah, it's the levels of hormones
Oh,
which will direct the behavior of that seed.
okay.
And of course, the conditions may vary across with the species.
Okay.
Yeah. So there's also embryo domains, which is non-hormonal, but it's controlled by chemicals. Later on, when we talk about these chemicals, these are adenochemicals, because adenochemicals can suppress germination. We have phenolics, unsaturated lactones, short chain fatty acids, coumarin. These have been proposed as germination inhibitors. within certain species. And in order to facilitate germination, or in order to break dormancy, leaching and oxidative destruction of these chemicals within the seed would be a requirement for breaking the seed dormancy. Later on, in a completely different topic, we shall explore these chemicals when we discuss adenopathy. Now, we are going to explore the coat-imposed domains. This is the outside covering of the seed, which may restrict the growth of the seed. So, seed domains in many species is imposed by the structures surrounding the seed. These structures include the seed coat or tester, pericarp, and the glumes, palia and lemma in cereals. For example, in Avena fatua, the palia, lemma, and pericarp are responsible for coat-imposed domains. But anyway, to simplify this issue, we can just say it's the outside coat which can restrict the germination process. And this brings about seed dormancy or coat-imposed dormancy. You can see this is the seed of Avena fatua, wild oat, which is quite hairy. You can see it may be difficult for water to penetrate this seed, or even gases like oxygen, which are required for the germination process. It's just waterproof, you know. So this is the seed. Here again, we have the seed of Avena fatua showing various parts. What I want to emphasize is that it may be difficult for water to enter these seeds, to go through the seed coat. If you look at the structure of the seed coat, where you have the pericarp, seed coat, then endosperm, this barrier around here is very difficult to break. So it's quite waterproof. It's a waterproof. And it may not also allow gases to enter. So this may cause dormants. It can also happen in Acanthospermum hispidum, where you have this seed coat, which may not allow water to pass through, or even probably some gases to pass through. So, with respect to the germination process, it may not occur, causing dormants. Remember, we also talked about immature embryo. It can also play its role. So there's immature embryo, there's the seed coat, which makes the seeds difficult to germinate. So let us look at the coat-imposed domains. How do the seed coat structures prevent embryo germination? Number one, the seed coat may prevent water uptake. If there's no water, there's no germination. The seed coat may prevent gaseous exchange. If there's no oxygen, there's no respiration, there's no germination. Uh, the seed coat may contain chemical inhibitors in the seed cover that may also stop the germination process. Of course, depending on the weed species. Uh, Preventing leaching, that is the seed coat, it may prevent leaching of inhibitors from the seed. Now, the seed may contain inhibitors inside it, and you may want to get rid of them before the seed can germinate. If the seed coat cannot allow leaching process, then there's dormancy. The seed coat may also alter light, which strikes the seed, and modify that light in such a way that the light is blocked from penetrating the embryo, and this can cause dormancy. The other issue could be just a physical prevention of the germination process. It's a mechanical constraint, this seed coat. It cannot allow the plumule to get to break it. Now, seeds with seed coat imposed dormancy are sometimes called hard seeds, where the seed coat is playing an important role. We have got plant families where we find hard seeds. We have got the leguminosae, the Canopodiaceae, Malvaceae, and Solanaceae families. These exhibit seed coat imposed dormancy associated with the prevention of water entry in the seed. Now, on coat imposed domains, you may have the wax cuticle, which can be a major impermeable barrier to water entry, where you have polyphenols and lignifications. These act like a waterproof, and you have your embryo, which is inside, and the water may not actually penetrate this barrier. Some seed coats may prevent gaseous exchange. Such seeds are said to be gas hard. For example, Xanthium species. You know, we've been talking about Xanthium possessing two different types of seeds. The seeds could also be gas hard. It may not be easy for gases to penetrate those seeds. So, impermeability to oxygen may be due to the presence of mucilages in and around the seed coat, or the consumption of oxygen by the seed coat itself. What may happen is that oxygen can be absorbed by the seed coat, and it may be prevented from entering the embryo. So let us look at the mechanism of this coat imposed dormancy Anyway, we are continuing with this explanation on court seed court imposed dormancy. So you may have insufficient availability of oxygen to support respiration for the germination process, or oxygen may not be sufficient to oxidize the inhibitors, which have to be broken first prior to the germination process. Please remember that the germination inhibitors are actually located in the embryo cotyledons, endosperm, or seed coat. That is where you may find the inhibitors. Some inhibitors may be found inside the seed, and the outer coverings may prevent leaching of these inhibitors. So sometimes when you soak these seeds, the inhibitors will remain inside, and therefore the germination process is not going to take place. Thus, assuming the seed dormancy status. Now, when the embryo is isolated and put in water, the inhibitor is leached out and the germination occurs. For example, in wild oat, Avena fatua, and also Xanthium species. Now, I wanted to move on and talk about light requiring seeds, which are said to be photoblastic. Now, what really controls the germination process is the substance known as phytochrome, which is found in the seeds. The ratio of the active phytochrome, which is known as far red, PFR, and the inactive phytochrome, red, which is simply PR, determines whether the seed is dormant or not. So it's the ratio of these two types of hormones inside the seed. This ratio can actually be changed by the light quality which shines on the seeds. So depending on the flash of light which is targeted to the seeds, it can actually determine how that seed is going to behave. The optimum ratio is established after exposure of the seed to white light, which converts PR to PFR in the embryo. I think this is what can cause the seed to germinate. The outer coverings form a filter to high incident light, and the seed eh, on the seed, and actually modify its effectiveness in converting it from PR to PFR. Remember, it is the PFR which is required to cause germination. This is the active phytochrome. So the process will be slow. If you have these seed coats, they will alter this process. So the perception of light by the seeds is dependent on the coats, and this can actually cause dormancy. We have a Canopodium album 30N, which has two types of seeds. Seeds with the dark seed coats, which are less responsive to light than their thin light coated counterparts. So you have dark seeds and the white seeds. Dark seeds are not stimulated by light, white seeds are stimulated by the light. Dark seeds filter out light and reduce the conversion now of PR to PFR when exposed to light. Hence, they remain dormant for longer periods than light coated seeds. I will show you the seeds. So, this one can easily convert uh, the inactive form of phytochrome to the active form of phytochrome when exposed to the white light, to the light. But the dark coated seeds, these will take time to convert PR to PFR. So they will remain dormant. Same seeds from the same plant. So the plant family is the Canopodiaceae, Canopodium album. Well, I wanted to now talk about the genetic control of innate domains because we said some seeds, when they come out of the plant, from the plant, sorry, they already possess innate domains. And we are saying it's under genetic control. So innate domains appears to be under simple genetic control, but sometimes the genetic control is modified by maternal effects. We have got examples. Nicandra physaloides possesses one pair of isochromosomes. The presence or absence of the isochromosome now determines whether a seed will germinate readily or is dormant. The seeds which germinate readily, you've got 2n equals to 20. These are the number of chromosomes. If the number of chromosomes happen to be 19 in some of the plants, it means that those plants will produce seeds which are dormant. Anyway, these isochromosomes are like this. Those with the 2n equals to 19, they have this arm is absent. And where you have chromosomes like this, you have 2n equals to 20. And probably germination can easily take place here rather than here. So this is from inside the plant. It's genetics, which is now at play. There's also an interesting plant known as Spegula avensis. We can associate its seed coat character, which is genetically controlled. Uh, we can associate this seed coat to seed to seed germination. So there are three seed forms. Homozygous papillate. A papillate means the seed has got some sort of bristles or hairs. And these structures, which are called the papillae, are 120 in a homozygous condition. And we can say the homozygous coat is smooth form, a smooth coated form, and also a heterozygous form, which bears 60 papillae. So the differences in the numbers of that papillae, which is genetically controlled, uh, may have some impact on the germination process. For example, the papillate seeds germinate readily at 21 degrees Celsius, while the reverse is true at lower temperatures. So in this case, uh, it's the genes which are controlling germination. This is also an example of genetic polymorphism, which is the production of seed of divergent morphology and behavior, but as a result of genetic segregation. Now, apart from the genetics, there's also what we call somatic polymorphism, which can affect innate domains. Now, with the somatic polymorphism, you may have you may have seeds which behave differently as a result of being produced on different parts of the plant. So, somatic polymorphism is the production of seeds of different morphologies or behavior on different parts of the same plant, not as a result of genetic segregation, but of somatic differentiation. We have got an example of Xanthium species. We also talked about it, where we have two seeds which are produced in a single dispersal unit. One deeply dormant, this is the upper seed, and the lower seed is less dormant. This was reported by Beverly and Blake in 1982. Now, the domains breaking requirements of these two seeds are different, with the result that at least 12 months normally separate the germination of the two seeds. So, what it means is that the seeds of the same plant, some may germinate readily, but others may have to wait for 12 months in order to germinate. Avena fatua may also give an example of somatic polymorphism, where you have seeds on different parts of the plant behaving differently. In Avena fatua, the grains on different positions on the spikelet have different germination requirements, according to Harper 1977. Polymorphism is important in separating the germination of the seed from the same plant over time. I think this is a survival mechanism. where you don't have weed seeds germinating at the same time. Otherwise, they will be destroyed during weed control measures. So some have to escape these measures through this mechanism. Polymorphism is widespread in the composite cruciferaceae and Chenopodiaceae families. These are the plant families where we get the weed species. Now, what we have been talking about for quite some time is the innate domains, which is quite a big subject. Now, we're going to talk about the other two types of domains. For example, this induced domains. This happens as a result of sudden physiological change due to changes in temperature, moisture, and oxygen levels. Now, what may happen is that if the conditions for germination are not optimum, the seed may not germinate, but something can happen inside the seed that keeps the memory of the outside conditions. So that memory which is inside the seed will cause it not to germinate. It could be some biochemical reactions which may take place inside that seed. So let's suppose we create good conditions for germination. Dormancy will persist because something inside the seed is telling the seed not to germinate. For example, seeds buried by tillage will undergo dormancy. Now, if you bring these seeds to the soil surface where there's light, good conditions for germination, they will fail to germinate. They still have the information which they acquired when they were buried in the first place. It is still there inside the seed. That's why the seed is not germinating. So this is induced domains. There's another domain which is more or less like this, which is governed by environmental conditions. This is
Hello?
hello.
I thought you paused your video.
Okay, it's okay. Right. The
Ah,
now the enforced domains. Now this can be due to placement of weed seed deeper than 5 centimeters, resulting from probably tillage. It is caused by the absence of red light under the soil surface. We know red light induces the germination in seeds by activating their phytochroma system, which is chromophore blue pigment attached to the protein molecule in the seeds. These are the details of the phytochroma probably structure. But what is important is that what senses light is the phytochrome inside the seed. The far red light deactivates the system and thus induced domains in weeds. So what happens if you bury the weed seeds? You limit far red, light. Sorry, let me answer this one. Hello. Yeah, those of Onaga. Okay. So the far red light deactivates the system and thus induces dormancy uh, in weeds. So dormancy does not persist when the environment changes. So when you bring the seeds to the soil surface, they normally germinate. They can only suspend the germination process as long as they are buried. But once you change the condition, the germination process takes place. But if those weed seeds refuse to germinate, then that becomes induced domains. So enforced domains, this is temporary, suspension of germination. Whereas induced, it's more or less permanent. Although later on seeds may come out of it. Here I'm now just going to explore environmental conditions that cause or terminate weed seed domains. Now, what can actually terminate domains? It could be light. Light causes weed seed dormancy. Some weed seeds require light in order to germinate. For example, garden soga paviflora, Portulaca orera C, Canopodium arbum, Amaranthus species. Breaking domains is related to light, which exists in two forms: a promoting and inhibiting form. The promoting form favored by red light that can cause germination, and the inhibitor form, which is far red light, which can stop the germination. Probably let us look in detail a bit about light effects. Inside the seed, you have the inactive form of phytochrome, which can be PR. If you flash that seed with red light, the germination actually is promoted. Now, when you have the active form of phytochrome, PFR, which can actually allow germination, if you flash those seeds with the far red light, you are now reducing the germination, and there's germination inhibition. You cause dormants. Now, using this principle, we can actually control weeds easily. For example, if we have fast growing plants which can form leaf canopy and alter the light from far, alter the light, promoting far red light, this can inhibit weed seed germination. One of the strategies which is used in conservation agriculture is the use of mulch, which can actually prevent light from reaching the seeds. And as a result, you can promote dormants and the seeds will fail to germinate. But of course, the drawback is that most farmers may not get enough mulch. But Maroch can work on this principle of light alteration, promoting far red light and probably darkness, eliminating light instead of stopping seed germination. Immature embryo, presence of immature embryo. Some weeds shed seeds with immature embryos. So seed storage may be required for several weeks before you can get the germination process. So we should be aware that some seeds may not germinate because of the immature embryos. We talked about the impermeable seed coat. Seed coats may be impermeable to water and oxygen. These are hard seeds which are found in Amaranthus species. The seed coat could be changed by scarification, action of acids or microbes. Now, the hard seed coat can prevent the germination process because the radicle cannot penetrate it. It's a mechanical prevention. Now, if we want to promote germination when we are having hard seeds, we may do scarification by rubbing them using sandpaper or pricking them with pins. Now, the issue of inhibitors, seeds are shed with some inhibitors, and they may not germinate. So, leaching may be required. We have to leach the germination inhibitors to improve weed seed germination. Please note that there could be external inhibitors like aerochemicals, from aeropathy. We may have to talk about this at some point. Then oxygen, low oxygen or low concentration levels of oxygen can also reduce germination in the soil. Temperature is important. We have got minimum temperature where no germination can take place, maximum temperature where no germination can occur. However, we should have optimum temperatures which are required by each weed species. So this can vary among weed species. For example, Amaranthus species will remain dormant at 20 degrees Celsius for up to 6 years. So raising the temperature would actually break dormants. Now, after ripening requirement, we talked about it. This is necessary for some weed seeds. The embryo is fully developed but will not germinate even if the environmental conditions are suitable. Germination will only occur when the seed is ripened. So there are certain processes which are occurring inside the embryo. Although it looks like it's a mature seed, but some processes are still going on and they prevent the germination process. So this has been quite a bit of information just to guide you You may have to read to use this summary. Just remember, our focus is on the seed behavior, on the weed seed behavior. And we said that one of the characteristics of weed seeds is the seed domains, which can be divided into primary domains and secondary domains. And the primary domains is the innate domains. That is the domains which is found in seeds which are being shed by the weeds. Then the secondary domains is actually acquired in the field. It may be induced or enforced. So there are three types of domains, which is innate, induced, and enforced. We talked at length on the innate domains. We can simply summarize it like this. The first issue under innate domains is what we call embryo domains. Now, when we talk about embryo domains, there are three things. The first one is immature embryo, which can be found in Acanthospermum hispidum or Xanthium stomarium. Second, it could be the issue of growth regulators, presence of ethylene in Avena fatua, gibberellins, Ga, and abscisic acid, Aba. We now know what they do in regulating the germination process. Or it could be external growth inhibitors, or they could be internal. But these are not hormonal, or they are not growth regulators. Things like coumarin and phenolics. This is under embryo dormancy. Under seed coat dormancy, we said the seed coat can prevent, number one, water from entering the seeds. It can prevent oxygen entering the seeds. It can restrict embryo growth mechanically, or it can possess growth inhibitors. It can prevent growth inhibitors from leaving the seed, or it could be alteration of light characteristics which affect dormancy. Then we talked about genetic polymorphism. For example, the presence of isochromosomes in the seeds of Nicandra physaloids, which can account for dormancy. Another weed where you have a smooth and rough textured seed coats, which is associated with the dormancy, and this is controlled by genes, which can control the smoothness or the roughness of the seeds. Then there's also something known as somatic polymorphism, where seeds are produced from different parts of the plant and they behave differently. For example, Xanthium stomarium produces two types of seeds with different domains characteristics. Also, Avena fatua seed can produce a seed on different position of the spikelet, and they have different germination requirements, which actually accounts for the seed dormancy which exist in these species. After that innate dormancy story, we talked about induced dormancy, and we said that the wheat seed is exposed to environmental conditions. It could be cold conditions. Then something inside the seed happens, and the seed will have a memory of cold conditions. Even if you create good conditions, you raise the temperature, it's not going to germinate because of the physiological changes which have taken place inside the seed. So this one will take quite a while to break this kind of induced dormancy. But the enforced dormancy is easily reversible. You bury with the seeds, they go into dormancy. You plow, you bring them on the soil surface, you break the dormancy very easily. This is enforced dormancy. Now, how do we terminate dormancy? Probably under laboratory conditions. We can use stratification, uh, maybe using hard objects to damage the seed coat, stratification, freezing the seeds, temperature, varying temperatures, use hormones, gibberellic acid, potassium nitrate. There could be other substances which can be used to terminate dormants. So remember that this trait or this characteristic of weed seeds, which is known as dormancy, regulates the emergence of weed seedlings under filmed conditions. That is probably why seeds, weed species are persistent in agricultural fields. So this is the end of this lecture. It's quite a long lecture, actually. We have to stop.